Don't use a 90, 80, 70, 60 scale. The performance on the exam I was pretty pleased with. Okay, so overall average was 71. The low score was 25. The high score was 98. So pretty wide range. And what I was pleased about was that there were a lot of scores in the middle range. There weren't a lot of scores at the bottom. So that makes me feel better. I wish I obviously want to see grades go up. But overall, I thought it was a pretty good performance on a first exam. All the exams we'll have in the course this term will be in the same format. So there's no adjustment to make from one exam to the next. You'll see the same format in other exams. And as always, if you have questions or anything, please come see me. I will post a key outside of my office. My office is ALS 2145, and that key will go up in the morning. Again, I can't do it tonight, but I will get everything by morning. You should have a key. You should have grade distribution. And if you have any questions or anything, let me know. Yes?
Will you post the key on Blackboard, or will it only be outside your office?
I will only post it outside my office. That way you have to come in. That way you get to know where my office is. What's that? ALS 2145. Okay? And that way, if you have questions, you can stick your head around the door and say, hey, Kevin, I have a question. That also makes it easy. Okay? Okay. Other questions or comments? Everybody got their exam? Okay. So I want to finish up today talking about membranes, and then we'll start talking about nucleic acids. So last time I got you going on this very important pump, and this very important pump, as I noted, was pretty critical for life. This pump allows a cell to sort of play with osmotic balances, and in playing with osmotic balances, it keeps the cell from bursting and getting full of water. Very important thing. As one of the students in here yesterday noted, it also creates a charge gradient. A charge gradient. So charge gradients also can give us energy if we use them properly. All right? The charge gradient is, remember that for each time this thing goes around, 3 sodiums are going out, 2 potassiums are coming in, and that means that each time it goes around the cycle, we're creating a net gain of 1 charge outside of the cell. Okay? So this is a really important thing. I don't care that we learn the mechanism here. The mechanism is not important, but what this pump is doing is very, very important. You'll notice I use the term pump, and I like to use the term pump when I'm talking about a protein that's involved in active transport. Just like a pump pumps water, it requires energy to pump water, so too does a pump that is doing active transport require energy. So that's an important consideration for us. Okay. I told you on the last one that the last pump used energy from ATP. Okay? This guy that you see on the screen does not, in fact, use energy from ATP to make it work. So we have to learn a little bit about how this pump is working. This pump requires a second pump to do its thing. Okay? So the pump that we're interested in here is the lactose. It's called the lactose permease. It moves lactose against a concentration gradient. So it's moving lactose from outside the cell where the concentration is low to inside the cell where the concentration is relatively high. Okay? Bacteria have this pump. Well, this pump does not use energy from ATP to move lactose against a concentration gradient. Instead, it uses what's called a proton gradient in order to make this happen. Okay? You notice it says galactoside permease, same name. So they're equivalent. Lactose permease, galactoside permease. Okay. So that means if this pump is going to work, we have to have more protons out than in, because the protons are going to want in, and they're going to be a driving force to bring in lactose. Well, how do we get protons out? Well, the bacteria have this. They have a proton pump that facilitates this happening. Okay? They kick protons outside of the cell. The proton concentration out here is high. They want in. So when they come in, they basically carry with them a lactose. That brings lactose against a concentration gradient and makes this an active transport system. Okay? So very, very important consideration right there. This is a prime example of a pump that does not require energy from ATP. So not all active transport requires ATP energy. Okay. Now, the last thing I want to say has really nothing to do with pumps, but it has to do with other ways of getting things across cell membranes. You'll remember when I talked yesterday, I talked about how we have, for example, water moves across a membrane fairly readily. Carbon dioxide moves across a membrane fairly readily. All right. We talked about proteins, that some of these pumps that are involved. We talked about the lactose, the gluts that let glucose in, and various things. All right. But there's one other way that things get across membranes that's significant that we should understand, and that's what you see on the screen. Okay. This mechanism that you see on the screen is the way that our cells take in cholesterol and other nutrients. This is how they take in cholesterol and other nutrients. All right? It doesn't involve a pump, so I need to explain to you what it's doing. I hope I've convinced you that cholesterol is important for cells. They use it in their membranes, and it gives those membranes some characteristics that we don't fully understand, but some characteristics that appear to be important for those cells. Well, cholesterol in your body is a very nonpolar substance. It's a lipid, right? And so it doesn't like the aqueous environment of the bloodstream. It doesn't like the aqueous environment of your cells. And just like fat, cells have to have special ways of handling it. Well, in the case of cholesterol, and we'll talk a little bit about this later, what cholesterol does is it's carried in your body by what are called lipoprotein complexes. Lipo, L-I-P-O, protein complexes. And what these complexes are basically proteins that are quite a few of them, and they wrap the cholesterol. So on the inside, the cholesterol is associating with the nonpolar portions of these proteins. The proteins then on the outside have hydrophilic groups that interact with water. So kind of like a grease droplet can be encased in a bubble of soap, cholesterol gets encased in a bubble of lipoproteins. Fat is moved in the body in a very similar fashion. In fact, in the very same fashion, the very same complexes that move cholesterol move fat. Fat-soluble vitamins are moved by these complexes as well. So these complexes play very important roles in carrying hydrophilic lipid substances from one part of the body to another. Well, the way, if they just carried it from one part of the body to another, that wouldn't do any good, because the cells want cholesterol, the cells want fat, the cells want fat-soluble vitamins. Okay? So what happens is, to get the things into the cell, there is a process called receptor-mediated endocytosis. Big mouthful of a name. I'll say it again. Receptor-mediated endocytosis. Endocytosis is E-N-D-O-C-Y-T-O-S-I-S. Receptor-mediated endocytosis. That's the process that you see on the screen that I'm going to just say a couple words about. Okay? LDLs are carrying this cholesterol. They're carrying this fat. They're carrying these fat-soluble vitamins. They're floating around your bloodstream, and they need to get into a cell. Your cells want to get this stuff, and so they have on their surface things called LDL receptors. LDL receptors. So these are various proteins on the surface of cells that will, as you see here, bind to an LDL. They will then cause this place of the cell membrane where they're located to pinch off and internalize that LDL. So the cell now has gotten the cholesterol, the fat, all the goodies that it wanted inside of here, and then it's simply a matter of opening it up and taking all the pieces out. You've gotten the cookie in, now we're going to eat the Oreo inside. That's basically what the cell is doing. It's going to use that fat, fat-soluble vitamins, and cholesterol. Well, this process turns out to be very important in controlling the levels of cholesterol that we have in our body. So many of you, if you've ever had a cholesterol check, probably have heard of LDLs. You hear of LDLs described as bad cholesterol because they're fairly loaded with cholesterol, number one. Too high of a level of these is linked to atherosclerosis. And we'll talk about why some people have high levels later in the term. Okay? But this is how they get into the cell. They play a very important role in our bodies because without these, we don't have a way of getting these things into our individual cells. Make sense? Clear as mud? Is anybody awake? Nobody's awake. Who knows a joke? How do you what? I have a feeling I don't want to know this joke. So you got two deans sitting on an airplane. You know, I can make fun of deans, right? Hi, deans. Right? Deans aren't known as being the smartest people in the world. Oh boy, now I'm in trouble. This will be all over the internet, right? No, I'm joking. But all right, so two deans sitting on an airplane, right? And they're flying on a red eye, right? And so they're going from New York to Los Angeles. And the pilot comes on and goes, "Hello, this is doctor. This is your pilot speaking. We're flying from New York to Los Angeles, and our estimated flying time is 4 hours. We'll be arriving in Los Angeles at about midnight. Not quite a red eye, but we'll be arriving at about midnight." I said, so they kick back, you know, and they lower the seats, and they're just sitting there, and all of a sudden there's this boom, you know, and the one dean looks at the other and he says, "What was that?" "I don't know." The pilot comes on and goes, "Whoa," he says, "sorry about that." He says, "You know, we're having a little engine problem here." He says, "We lost number 4 engine, but, you know, don't worry, we have 3 engines on this plane. It'll only delay our arrival in Los Angeles by 1 hour, so we can expect to arrive in Los Angeles at 1 a.m. Dean says, okay, you know, goes back to sleep, and they're laying there, you know, and pretty soon there's boom! There's a bigger boom, so voice went up on that one, okay? Pilot comes back on, and he says, "Well," he says, "you're not going to believe this," but he says, "You know, we've lost number 3 engine," and he says, "But we still have 2 engines," and he says, "It's only going to delay our flight by another hour or so, so we should be arriving in Los Angeles by about 2 a.m. "Yeah, okay, you know, back to sleep. You know what's going to happen next, right? Boom! Pilot comes on and goes, "I can't believe this. I've never been on a plane like this before." He says, "You know, we've lost number 2 engine." He says, "We've only got 1 engine left," but he says, "It's a pretty reliable engine." He says, "You know, we should be arriving at Los Angeles by," what time is it now? 3 a.m. One dean looks at the other and he says, "Man," he says, "if that last engine goes out, we're going to be flying all night." He's a dean, you see. Okay, that was the best one I had. You see, never mind. Okay, so yesterday I told you about the fat-soluble vitamins. The last thing I want to talk about here are an interesting class of compounds, the last of the lipids, called prostaglandins. Prostaglandins, leukotrienes, and thromboxanes, okay? This is what they look like, okay? Now, we'll talk later about how they're made. They're actually made from fatty acids. You may have noticed the other day that one of the fatty acids was called arachidic acid, and it had 20 carbons. You probably didn't notice that. But if you did, you would see that this looks very much like it. And this guy right here is made in an interesting reaction. I'm sorry, this is the starting material. This interesting reaction converts this guy here into what are called prostaglandins. Well, what's happened? What's happened is that arachidonic acid has gotten a joined bond right here, and it's made something that we call a cyclic compound. This is just basically a bent compound. This guy's cyclic because it has a ring in it. Okay? Now, I'm not going to tell you how that's made right now, how it's made and what affects it. Aspirin, for example, controls whether or not you make these. Okay? These guys cause pain, and aspirin inhibits their production, so aspirin's a pain reliever. Kind of cool, right? these guys right here play very important roles, many very interesting roles in the body. One role that prostaglandins play involves pain. So if you get stung by a bee, for example, prostaglandins may be made in abundance, and one of the reasons a bee sting hurts a lot is because of that. Your headache. Your headache is involving prostaglandins. You take pain relievers to stop prostaglandins from being made. Prostaglandins also are involved in things like blood clotting. So when we think about people taking aspirin to keep their blood thin, well, it's not really thinning the blood, but it's preventing the production of certain prostaglandins that are involved in clotting. So they're reducing the likelihood that clotting is going to occur. Okay? Some prostaglandins are involved in uterine contractions during birth, childbirth. Okay? So prostaglandins play some very, very important roles, and that's what they look like.
So,
Yes?
does the prostaglandin actually cause the pain, or?
Okay. So prostaglandin is produced, and the production of that results in stimulation of nerve cells that are involved in pain. So yes, in a sense, you could say it does cause the pain. So if you prevent the production of prostaglandins, then you reduce the likelihood of pain. Make sense?
Yeah.
Sure. Yeah.
Prostaglandin is causing the pain, does it also cause the
When it's causing the pain, does it also cause a contraction? Prostaglandins they're very interesting compounds. Prostaglandins tend to have very specialized functions, so there's actually quite a wide variety of them. So the ones that are involved in uterine contractions are not directly involved in pain, to my knowledge. Make sense? Yeah. Yeah. I'm sorry, say it again?
Allergies? Like, do they combine with allergies and you get stung by a bee?
Oh, that's a good question, actually. Do prostaglandins combine to make the allergies worse for people who are allergic to bee stings? To my knowledge, no. Most of the allergic reactions that happen with respect to bee stings and other things like that are actually produced by the immune system, not by the prostaglandins. But it may very well be that those prostaglandins being produced are actually recognized by the immune system. So I would say directly, no. Indirectly, probably. Yeah?
So that last diagram, are you showing that arachidonic acid is a precursor to creating a prostaglandin?
Yes. And we'll talk about how that's made later, so don't worry too much about that. But I just mainly want you to know what prostaglandins are. Okay? You notice I gave you three classes of compounds. I talked about prostaglandins. Now I want to show you a related compound called leukotrienes. And leukotrienes, and this is getting back to her question up here, have a sort of a similar structure. Okay? They have some odd things about them. But leukotrienes are involved in very strong allergic responses. Okay? And they may very well be implicated in things like asthma. Okay? So they're made in a similar process to prostaglandins, not exactly the same. And again, we're not worried about how they're made right now. We're just more interested in what they are, and we'll talk about how they're made later.
Is that leuko or leuko?
Yeah, it's L-E-U-K-O. It's right there, leukotriene. Okay? All right, and the last of these is thromboxane. Thromboxane, again, looks kind of like these guys. Thromboxane is actually made from prostaglandins. And the importance of these guys is thromboxanes are implicated in making blood platelets sticky. That's also an important consideration in blood clotting. So since thromboxanes are made from prostaglandins, if you stop the production of certain prostaglandins, you will stop the production of certain thromboxanes, and again, that's helping to make blood less likely to clot. This is going to be a very important consideration for people who have various issues with blood clotting. Maybe they've had a history of a heart attack or stroke or something like that. They may get put on to aspirin for that reason. Yeah?
So that makes blood cells sticky?
It makes blood platelets sticky, yes. Okay. Well, that's what I want to say about membranes. So move forward and talk a little bit about nucleic acids. All right? Everybody's heard about nucleic acids, right? So as I start talking about nucleic acids, I want to say a little bit about structure. There's some nomenclature that we'll talk about that I think you should be aware of. And we'll talk about function as well. Excuse me. Okay. So, nucleic acids are polymers of nucleotides. Okay, we said proteins were polymers of amino acids. Nucleic acids are polymers of nucleotides. The nucleic acids we have in our body, of course, are DNA and RNA, deoxyribonucleic acid, and ribonucleic acid. And before we talk about those, we need to understand something about nucleotides. So nucleotides are the building blocks of the nucleic acids, and nucleotides contain three components. Three components. You should know this now, okay? The three components we find in a nucleotide are a base. That's probably what you thought a nucleotide was. What you see on the screen are bases. Those are not nucleotides. These are bases. They're part of a nucleotide. Second component of a nucleotide is a sugar. In the case of RNA, the sugar is ribose. That's the ribonucleic acid. In the case of DNA, the sugar is deoxyribose, and that's why we get the deoxyribonucleic acid. So the two components so far of a nucleotide, the base, the sugar. The third component of a nucleotide is at least one phosphate. Okay? If all we have is a base and a sugar, we don't call it a nucleotide, we call it a nucleoside. Okay? Nucleoside. If we describe something as a nucleoside monophosphate, that means nucleotide, right? Because a nucleoside with a phosphate on it now has all three components. That would be a nucleotide. And you'll frequently hear that designation. A nucleoside monophosphate, a nucleoside diphosphate, a nucleoside triphosphate. ATP is a nucleoside triphosphate. Sorry. So take a nucleoside, put three phosphates on it, you have that nucleotide. Make sense? Well, if I said nucleoside plus phosphate, I call it a nucleotide, right? I can't call it a nucleotide phosphate because that would be redundant, right?
Okay, so you're just saying that there's
So-
more phosphates on it.
That's right. So her question is a good one. So her question that she said, if you didn't hear it, was, why do we call it a nucleoside then? And so we're trying to be more descriptive. A nucleoside monophosphate, or a nucleoside diphosphate, or a nucleoside triphosphate tells me something about structure, but all nucleotide tells me is it's something that's got at least one phosphate. Make sense? Yeah. Good question. Okay. So, you know, of course, I think everybody knows from basic biology that DNA has cytosine and thymine. RNA has cytosine and uracil. These guys are single-membered rings. They are pyrimidines. So pyrimidine is the big category. The big category for the other guys are called purines. And you see that they have two rings joined together, a 6-member and a 5-membered ring. Adenine, we find, of course, in both DNA and RNA. And guanine, we find in both DNA and RNA. Okay. Here's some nucleosides. All right. So there's a ribonucleoside. All right. Has ribose. There's the structure of ribose, and we'll talk about that later. But ribose is a 5-membered ring. Ribose versus deoxyribose, the only difference in the sugar is right there. Ribose looks like this, deoxyribose looks like this, and deoxyribose is missing an oxygen, hence the name deoxyribose. So that's what we have right here. Okay. All right, nucleotides, of course. You can see them up close and personal. You don't have to draw the structure of these. But I think you should be able to sketch out the general structure of a nucleotide, right? You could sketch a 5-membered ring. You could sketch a base above it. You could sketch a phosphate. You know, just something like that. I'm not going to ask you where all the carbons and nitrogens and all that sort of stuff are. But I think that's useful for you to know, just the general structure of those guys. In making a polymer of the nucleotides, what happens is an enzyme joins them together and creates what are called phosphodiester bonds. Phosphodiester diester bonds. All right, here's where your organic chemistry comes into play a little bit. There's the phospho part right there. There's an ester bond to one of the sugars up here. There's a there's an ester bond to the next sugar down here. You'll notice that the bases are not linked in this fashion. The bases are on the inside, away from all of this. And so if we think of a double helix, then we have one strand here, one strand here. The bases would be where my noses are. Okay? The bonds that we would have, pretty good visual, huh? The bonds would be along my fingers, right? So if we look at the outside of a nucleic acid, we see sugar, phosphate, sugar, phosphate, sugar, phosphate, right? And each of those sugar-phosphate bonds is a phosphodiester bond. Okay. There we go. There's obviously the structure of DNA shown very schematically. You saw when we drew proteins that we use ribbons to kind of guide our eyes. We'll use the same thing with DNA. There's the ribbon to guide our eyes. And instead of drawing all those carbons and nitrogens and everything, we simply put the bases on the inside. Now, when we look at this, we see G is, of course, guanine. Guanine was a purine. Purines were big. Bigs are always paired with small to make the same size. So if I have a GC base pair, the helix has the same width as if I have an AT base pair. The width doesn't change because of that. That's really kind of cool. What holds the bases together, why the reason complementarity works, is because the bases are held together by your friend and my friend that we talked about in proteins, the hydrogen bonds. So hydrogen bonds are what hold the helix together. I talked earlier in the term about why having weak bonds holding the helix together made sense, because the cell has to pull those strands apart and replicate them. Oh, one last thing. The bases are arranged, the strands are arranged in what we call an anti-parallel fashion. There are ends to each. One end is called the 5' end, the other end is called the 3' end, and you'll notice that if I follow the purple strand, it goes from 3' here all the way down to 5' here. And the complementary strand is oriented oppositely. It starts at 3 and moves up to 5. Okay? So that's what anti-parallel means. Schematically, it looks like this. All right? Opposite polarity, but inside base pairing occurring properly. Now, the structure of the double helix of DNA was discovered by, as you probably know, Watson and Crick back in 1953, using data they stole from a woman named Rosalind Franklin. She was a remarkable woman, and she did not get credit. I always like to tell the story about her a little briefly, if I can. Rosalind Franklin was probably, we recognize today, one of the most preeminent female biophysicists of her time. At the time she did this, she was the only person on the face of the earth who knew how to make DNA crystals. And making DNA crystals was essential to determining the structure. Nobody else had figured this out. Rosalind Franklin figured it out. And the story by which the story by which Watson and Crick got to steal the data is an interesting one, and it actually relates to the building that you're sitting in right here. So I'll tell you that story. The story is that there was, as you know, a lot of interest in determining the structure of DNA. Has anybody in here ever read In Search of the Double Helix? Very good book. If you want to learn about something about it, it's well written, and it's actually written by James Watson, who was involved in this, and he admitted the stealing. Okay? Admitted the stealing. So here's what happened. So they have this race to get the structure. Labs around the world are working to see if they can be the ones to discover the structure of DNA. One of the labs that was very much involved in that race was Linus Pauling. And Linus Pauling had previously won the Nobel Prize for discovering the alpha helix and the beta strands of proteins that you've already seen. So Linus Pauling is pretty convinced that DNA has a helix as well, but he doesn't know the nature, doesn't know the structure of the helix that, of course, we now know. And so he's working very furiously on this, and there's a conference coming up in England, where all the DNA people are getting together to share their data, to talk about structure, et cetera, et cetera. All right? So Linus Pauling, at that time, you had to have, to leave the U.S. and go to England, you had to have a visa. And Linus Pauling applied to get a visa to go to this meeting, and he was denied one by the U.S. State Department. Why did the U.S. State Department deny him a visa? Because Linus Pauling was running around the world arguing that we should not be testing nuclear weapons in the atmosphere, which is what was going on at the time. Around the world, people were testing nuclear weapons in the atmosphere. I'm old enough, I remember being told we can't go outside because it's going to rain and we're going to get radioactivity on us. I'm not kidding you. Okay? These things would blow all around the world. Well, we take it for granted now that we don't do that. And we don't do that because Linus Pauling was making that case. The U.S. government said he was a subversive. Refused to give him a visa to go to this meeting. Linus Pauling doesn't get to go to this meeting. Rosalind Franklin, Watson, and Crick are all in England, and Rosalind Franklin doesn't get to go to the meeting either. Her boss gets to go. She's working in someone else's lab. What does he do? He takes her data with him. They're at this meeting, and they're sitting around and being a good old boys club, and they're all talking about everything, and Watson and Crick learn that Rosalind Franklin's mentor has data. Can we see it? Can we see it? Sure. Doesn't ask her permission, lets them see it. They look at it and quickly realize what some critical function or critical features of the DNA structure were. They took that information back to them, and within a matter of a very short period of time, months, they solved the structure of DNA using her information. Okay? Now, there's a little bit more to the story than this. So first of all, you've seen Linus Pauling might, if he had been at this meeting, had a chance to have been involved in this. Linus Pauling actually came probably that close to a third unshared Nobel Prize. That was the stakes. He's the only person ever to get 2 unshared. He might have had a third. Now, the little accentuation mark on the end of this story was that Rosalind Franklin's mentor is a man named Maurice Wilkins. And when the Nobel Prize was given for the structure of DNA, it was given to Watson, Crick, and Wilkins. Now, you might say, well, that was really, really, really unfair because Rosalind Franklin should have gotten it, and it turns out Rosalind Franklin couldn't have gotten it because she died of a very rare ovarian cancer before the Nobel Prize was awarded. You can't win it posthumously. Okay? The thinking is that the reason that she died of it is because to determine structures of crystals, you have to shoot X-rays through things. And in the days that they were doing this, it was considered macho not to use a shield. And Rosalind Franklin did not use a shield. And she died of a very rare cancer. Very sad story. Anyway, so that's an interesting story. I think science is interesting in an historical perspective, and seeing the players in that sort of reveals it. So if you're interested in a good book, I recommend that book. Okay. Well, the structure of DNA discovered by Watson, Crick, and, oh, by the way, if you look in that electronic textbook I gave you, I wrote a limerick about this thing that you might discover it in there if you get looking through there, about what would be fairer in naming the paper in which this was published. So it's called Watson and Crick, but I think Rosalind Franklin, it deserves to be on there. So look at the little poem I've written in there. I think you'll enjoy that. All right, I've got to watch the time. All right, so the structure that Watson and Crick reported and got credit for is called the B form of DNA. The B form of DNA is the most predominant form of DNA in the cell. Probably occupies about 99% of the DNA in the cell has that structure. In the very same issue of Nature in which Watson and Crick published the structure of DNA, and by the way, that paper in Nature was a 1-page paper. I argue that that was the most condensed set of information ever published in the history of science, because when we look at the revolution that's happened since then, that came from 1-page paper. Okay? A remarkable thing. In that same issue of Nature in which Watson and Crick published the structure of bDNA, Rosalind Franklin published the structure of aDNA. People don't realize that, but it was true. Well, the reason that everybody looked at bDNA and Watson and Crick was because it turns out this is the predominant form, and this form at the time was thought not to be very significant. It wasn't very significant. Well, we now know it does have some significance in the cell. Why did they think it wasn't significant? Well, the only way she could make these crystals was if she completely dehydrated them. And DNA, of course, exists in an aqueous environment. So the thinking was that there must have been some chemical artifact that gave rise to this. So that's very nice, Rosalind. You've got a very nice structure. But this is what we're really interested in. Okay? Well, we now know that this A form, in fact, does occur inside of cells. And there's really two ways in which we see it. One is anything that involves double-stranded RNA, whether it's RNA to RNA or RNA to DNA, is in the A form. Okay? Anything that involves RNA to RNA or RNA to DNA is in the A form, not in the B form. So wherever there's an RNA involved, we'll see A form structure. We also see it in DNA, and the place where we see it appears in places where DNA appears to bend. Appears in places where DNA appears to bend. Okay? And bends turn out to be really important for DNA because, as I think I've mentioned in class before, if we take all the DNA out of a single cell of our body and stretch it end to end, we have 7 feet of DNA. Bending it, making it fit into cells probably is very, very, not probably, it is very important, and this may well play a role.
So it's the A form up to the bend, and the bend is B form?
That's a very good question. So generally it's thought that you have a B form, you may have some sequences that favor A, there's a bend, where that'll be A, and then it goes back to B. You're exactly right. The last form, and I'm not going to say much about this today, although I may mention more about it on Friday, is the Z form. The Z form at first was thought not even to be possible. And then in the late 1970s, a man named Alex Rich crystallized a DNA that was in the Z form. Now, why do they think it wasn't even possible? Well, it turns out when you take two strands and you put them together into a double helix, there's two ways in which they can coil. They're called left-handed and right-handed. Okay? Now, I can't show you here. If you want to come by my office and see, I'll show you with a phone cord what left-handed and right-handed means. But I'm going to try to show you with my fingers. Okay? So one way to coil them in DNA is to do this. Okay? The other way in which we can coil them is to do this. We can think of this almost clockwise versus counterclockwise going up. Okay? And it's not the same as turning it upside down. Because if you turn it upside down, it still has the same orientation. So there's two ways of putting them together, left-handed and right-handed. It turns out that A DNA and B DNA are both in the right-handed form. So people say, well, Z DNA has got to be a weirdo. How can DNA go from being in the right-handed form and then flip backwards and go in the left-handed form? That can't happen in the cell. And what they said was the same thing they said about Rosalind Franklin's thing about A DNA must be an artifact. Well, now we know, in fact, that Z DNA in the cell does form, and there are certain sequences of bases that favor its formation. Okay? Now, I'm not going to talk about those today. I will save that for another story that I'll tell you on Friday, but that's what zDNA, the structure of zDNA actually has. Okay, now, there's left versus right. Try to illustrate it, but I don't think it does a very good job, unfortunately. A phone cord is much easier for me to show it to you. Okay. Why don't we have a song to finish? What do you guys say? All right. So this is a song to the tune of, everybody know the Yellow Rose of Texas? Okay? All right. This one's about the mellow woes of testing. All right? The term is almost at an end, 10 weeks since it began. I worried how my grade was because I did not have a plan. My first exam went not so well, I got a 63. 'Twas just about the average score in biochemistry. I buckled down the second time, did not sow my wild oats. I downloaded the videos and took a ton of notes. I learned about free energy and delta G naught prime. My score increased by 7 points, a C+ grade was mine. I sang the songs I memorized, I played the MP3s. I learned the citrate cycle and I counted ATPs. I had electron transport down in all of complex V. I gasped when I saw my exam, it was a 93. So heading to the final stretch, I crammed my memory and came to class on sunny days for quizzing comedy. I packed a card with info and my brain almost burned out. 'Twas much to my delight I got the A I dreamed about. So here's the moral of the song: it doesn't pay to stew. If scores are not quite what you want and you don't have a clue, the answers get into your head when you know what to do. Watch videos, read highlights, and review. All right, guys. See you on Friday. What is it? Osborne Haley? Hi.
I have a question about the exam. Can I just come by?
Yeah, come by, and I'll get the key posted in the morning. Okay.
Norson?
Norsum. Okay.
Yeah, Robert Sickles?
Stickles. Okay.
Thank you.
All right. Hi. Okay.
Well, I guess it's kind of a couple people we're talking to. So,
Uh-huh.
in this one, I think understanding it as only have one pKa
Uh-huh.
rather than always have at least one pKa.
Okay. All right. Come talk to me about it. I'll be happy to consider it for you. How about that?
Okay.
Okay. How you doing? Did you sing?
What?
Did you know the song? Probably didn't know the song. It's an old song. I'm an old guy. So I hope you had a good day today.
Yeah.
Good. Any questions? In fact, I'll leave you my card. I think you've got my information. If you have any questions, give me a holler. I'll be happy to answer your questions for you.
Okay.
And good luck in your choice of schools. Okay. Take care. Hi.
Do you want me to write a request for a regrade if I think you just